All right, today's topic is, sorry, pardon the cough, xylem and phloem and how we're going to move things into and out of the tree. The plant, it needs materials, it needs minerals, it needs water in order to get the sugar it needs to grow. And so we're going to talk about the two main vascular tissues within a plant, the phloem and the xylem. Think of this as your veins, but your veins are going to carry everything. So you have one type of vein, really. Whereas in a plant, they have one thing that's going to carry only water and another type of vascular system that's going to carry everything else. So the first type of vascular tissue that we're going to talk about is xylem. So when we talk about the movement of water, which is what xylem is going to carry, we're going to talk about transpiration. There is something called the transpiration pole, and that's pulling water in from the roots of the plant up to the top and out then through the leaves. Transpiration is the loss of water vapor from the leaves and the stem of the plant. That water then has to be replaced, and it's replaced by pulling water in through the roots. So water generally moves into plants through their roots and upward through the vascular system and then out the stomata of the leaves. Stomata tends to be on the bottom side of the leaves, so it's protected from direct sunlight. It's also not interfering with photosynthesis. And these are essentially the pores of your leaf. So stomata, if you look up here at this picture, I always think it looks like Pac-Man lips. In stomata, it regulates the transpiration. So it's going to regulate how much water is lost through this transpiration pole. Generally, your stomata are found in the leaves, although in some plants there could be some in the stems. Generally, they are open during the day and closed at night. They are made of two elongated guard cells on either side, and these are attached to normal epidermis cells. Remember, epidermis is your skin. So if I say the epidermis of you, it's the outer layer of your skin. The epidermis of a plant is the outer layer of a plant. The two guard cells are not attached to each other, and that way they can open and allow water out or close and keep water in. They open and close based on Tuger pressure. They open when water is absorbed by the guard cells from its neighboring epidermis cell, and then at that point water can escape the plant through the pore. They close when enough water is lost that the cell becomes flaccid or limp. When it becomes limp, it closes again. Again, they said they tend to be open during the day and closed at night, but if during the day it's really, really hot or really, really dry out and the plant's losing too much water, these cells will close as well. So if you think about, back in the beginning of school, we had these really hot days, that would trigger the stomata to close. If you look at this image, you are going to have to be able to, on a drawing or on a micrograph slide like this, identify where the phloem is and where the xylem is. Know that the xylem is often towards the center of the stem, the center of the plant, and the phloem tends to be more outward, out by the edge of the leaves. This is going to make sense, right? So the xylem is transporting water, whereas the phloem is transporting everything else. It's transporting what is needed for photosynthesis in addition to water, and then it's taking the sugar that's created and it's moving it to the other parts of the plant. And so you want the phloem closest to where the photosynthesis is. Let me try that sentence again. You want the phloem closest to where the photosynthesis is going to occur. Try and say close in photosynthesis like 10 times fast. It's not working for me. So make sure that you can identify that the xylem is often on the inside and the phloem's on the outside. So the xylem, water transport occurs in the xylem. It is one directional. It goes from the roots, up the stem, out the leaves. It begins as elongated cells that are connected end to end, and then the end pieces dissolve away, so you have essentially a straw that goes down the stem of the tree. Living tissues of xylem lay down cellulose. Cellulose is thickened by lignin. Lignin then becomes the woody part of the tree. So as a tree grows in girth, it's creating more and more xylem, and that center part becomes now the structural support for a woody tree. And when you look at the rings of a tree, you're counting the rows of xylem, essentially, that make that darker line. Again, you're going to have to know which is which. Your xylem is towards the center and your phloem is on the outside. This whole circular image right here, this is a vascular bundle. So it's got xylem here with the pink, and then your phloem out on the outer ring of this vascular bundle. Your transpiration stream is also known in scientific speak as the cohesion tension theory of fluid movement. That's like a mouthful. It's basically saying that you have a straw that sucks up at the roots and then comes out at the leaves. So as water is lost by transpiration, the osmotic pressure in the cells causes water uptake from surrounding cells in the xylem. As water leaves the xylem, more is pulled up through transpiration pull and the properties of cohesion and adhesion, so the water sticking to itself and the water sticking to the sides of the xylem. And then as the water leaves the xylem, more is pulled in from the root. Much of the water pulled into the leaves isn't used for photosynthesis. A lot of that water is lost as tree pee through transpiration. If you've ever sat underneath a tree on a really hot day and you've had drops of water falling on you, it's not raining. It's the transpiration, it's the water coming out of the stomata on the underside of the leaves of the tree. Certain things will increase or decrease the rate of transpiration or the rate of flow in that transpiration flow. The first one is light. So during the day, the stomata tend to be open. This increases transpiration because if the pore is open, there's more water being lost, which means you need to have more water being brought up from the roots. Temperature causes evaporation of the water molecules from the surface cells of the plant. And if you have a lot of evaporation, you increase transpiration pull, you increase this flow in the xylem. Evaporation rate doubles for every 10-degree increase in temperature above what that plant is evolved to deal with. When you think about wind, on a windy day, that water is being blown away from the stomata, and when the water is blown away, another drop replaces it. And so if you have high wind, you're going to increase transpiration. Okay, decreasing transpiration would be humid air. So we're in the middle of the monsoonal flow. I'm recording this in the first week of school, and there's a lot of water in the air, water vapor. So our humidity is sitting around 45%. Because that water is there, it's not allowing evaporation of the water at the pores of the stomata, and so you're going to decrease transpiration. Soil water, the amount of intake by roots matches the transpiration rate. High levels of carbon dioxide decrease transpiration because the stomata are going to close. So things that increase transpiration: light, temperature, and wind. Things that decrease transpiration are humid air and high levels of carbon dioxide. Your root system, your roots are huge, they're ever growing. For the entire history of the life of that tree, your roots are going to continue to grow. And this is why if you plant a tree in your backyard by your patio, eventually the roots will lift up the concrete. It's going to continue as the tree gets bigger to look for new and better sources of water. The surface area is increased not only by the length of your roots and the depths at which your roots grow, but also by the hairs on the roots. So if you look at this picture here, there's all these hairs that are coming off. That's increasing the surface area. The more surface area you have, then the more water uptake you can have. Cortex cells also have a structure that facilitates the uptake of water. Obviously, the quicker you can move water away from your roots, then you're changing the osmotic pressure and more water is going to rush in. In addition to water, the plants are going to get a lot of minerals and nutrients from the soil. And there are two ways that they're going to get those minerals in. They're going to get it in through diffusion. So as the plant uses the phloem to move the ion away from the root, it changes the rate, right? And in diffusion, things are always going to move from highest concentration to lowest. And if the lowest is in the root, then those ions will diffuse into the root to be utilized by the plant. The other way we're going to get this stuff in is mass flow of water. So some of these ions and some of these minerals are small enough that when water comes into the cell, it also brings with it the dissolved ions and minerals. The ones that we need for healthy plants are sodium, potassium, and calcium, and nitrate. So if you were to ever buy a fertilizer at the nursery, you want to have a fertilizer that has a ratio of sodium, potassium, and phosphate, as well as calcium. And different plants, because they've adapted to different areas, are going to ask for different things. So that's why fertilizer, say like great for fruits, great for cactus, great for vegetables. And you want to make sure that you're feeding that plant what it needs in the correct proportions. In addition to diffusion and mass uptake of minerals through water, we're going to have a symbiotic relationship in a lot of plants with fungal hyphae. These fungal hyphae are going to absorb the nutrients and they grow into the root itself. And so the fungus absorbs the ions and then passes the ions to the plant. So that's what the plant is getting from the fungal hyphae. The fungal hyphae are getting sugars from the plant. So it's a mutualistic symbiotic relationship between the two. If we have things that are too big to come in through diffusion, if we have things that aren't moving away from the roots very quickly, we could also have active transport of our minerals. In active transport, we're using ATP to move things through passages or gates, just like the ATP synthase that we're going to see later on in your neurons. So this occurs against the concentration gradient. Cortisol of the cell holds some reserve of ions, and it allows the cells to pull in more of these ions when available. So in other words, if you fertilize the plant might go, oh my gosh, potassium, I haven't seen potassium in forever. And so I'm going to hoard potassium, and I'm going to pull in more than I need right now. And because I'm pulling in more than I need right now, it's going to hang out in the roots, which means there's going to be more potassium in the roots than in the surrounding soil, and so diffusion doesn't work anymore. So we're going to have active transport pumping that into the roots. It's highly selective. If there are multiple forms of the same ion, if there's multiple forms of nitrogen, so if it's a nitrate, nitrite, nitrogen gas, the plant is going to choose the ion that it wants to use. It can use the other ions, the other forms in a pinch, but it's going to pick the one that is easiest for it to use first. If roots are deprived of oxygen, all active transport stops and the plants can't pull these ions in. Remember that the plants need oxygen in order to do cellular respiration. Cellular respiration creates ATP from the sugars that the plant made earlier in the day through photosynthesis. The sugars create ATP, and the ATP is used in active transport. Last thing I want to talk about in this unit are two specialty types of plants. We have xerophytes. Xerophytes are your desert plants. A lot of the plants in our area tend to be xerophytes. These plants are adapted to live in low humidity, high heat environments. So when we talk about a xerophyte, it's going to have a thick cuticle. This is going to be like a waxy covering on the cell, and it's going to prevent evaporation through the epidermis or through the skin. It's going to be hairy. So the hairs protect the plant from losing evaporation. And then if you've ever walked outside in the summer, in the early morning, there's a lot of dew. That dew can be collected on the hairs of these plants, and then the plants can absorb it from the leaves where it's collected. There's not going to be as many stomata. If we were back in the classroom and we had access to microscopes, there's this really cool lab where you take clear nail polish and you put it on the bottom of a leaf, and then you can peel it off and look and actually see the stomata. You're going to see rolled or folded leaves, anything that's going to reduce the surface area of that leaf, because if you reduce the surface area, you reduce evaporation and you reduce the need for water. There's going to be superficial roots. Again, this is going to be to take advantage of any dew, because you're not going to get rainfalls. You're not going to have a water table that's really accessible. So instead, you're going to have these superficial spread out roots that as the morning dew kind of soaks in, the plants can grab it. Some of these plants will have what's called a taproot that goes way deep down and it's searching for the water table. This water table could be 50, 60, 70 feet down underneath the ground. And so these deep extensive roots are looking for that. They're going to have a reduced number of leaves. Again, the less surface area, the less likely you're going to lose water to evaporation. And there's going to be limits to growth. So if you look at an oak tree in the desert, and sometimes we see oak trees growing where there might be water, these oak trees are going to be small and they're going to have really small leaves. And these oak trees could be 200, 300 years old and they're no taller than you are, as opposed to an oak tree in an area that gets a lot of water that in that same 200 and 300 years is going to be a 90, 100-foot tree. The opposite end of the spectrum here are halophytes. Halophytes are going to live in water. They tend to be in swampy, marshy areas. And they store water because they're in an environment, their roots, that have a high salt concentration. And so you need to store water to battle that salt concentration so you're not getting the ions in. They're going to have salt glands to expel excess salt. Mangroves are actually going to have like a salty crystalline along them on the top of them because they're getting rid of any salt that comes in with the water that they're pulling in. They have specialized vacuoles to store sodium and chloride. This prevents salt toxicity. They have protected stomata that are recessed in the cuticle. This increases the humidity and decreases transpiration, and they tend to have reduced surface area. Okay, that's it for xylem. Now we're going to talk about phloem. Xylem transports water, phloem transports sugars and minerals. So phloem tissue, we have sieve tubes, which are narrow, elongated cells connected to end with no real organelles in them. Again, it's like a straw. And the sieve plates are end walls which have pores to allow material. They have companion cells, which are going to maintain and feed the sieve tubes, and they're attached by cytoplasmic strands. Translocation is the movement of manufactured food. Well, what does that mean? It means it's the movement of the sugars that are created by photosynthesis by the leaves. But we don't want just that sugar going down to the roots. We also want that sugar going up so that the plant can grow bigger and taller as well. So phloem is going to transport things in any direction. So their sugars and their amino acids and their minerals are going to go in any direction of the plant. It's going to go up, it's going to go down, it's going to go sideways. Xylem differs in that xylem only goes in one direction. Xylem only goes from the roots to the leaves. Phloem flows anywhere. It's going to go from the source to the sink. A lot of times our source are the leaves where photosynthesis is occurring, and the sink is where it's going to be used. That sink could be the roots, it could be the shoots of the plant as it grows taller. It takes sugars to new growth in young plants and storage in older plants. When we think about storage, think about like a carrot that's growing underground that's going to store that sugar in that yummy, sugary carrot so that in times of winter, that carrot can grow from that stored sugar. Amino acids are produced in the roots and transported to where they're needed for protein synthesis. Again, usually the growing parts of the plant. And chemicals sprayed on the plant can be moved in this manner. So if I spray a pesticide on the leaves of my plant, it will move that pesticide down to the roots. We can measure the rate of translocation, the movement in phloem, in a couple of ways. And believe it or not, about 3 years ago, this aphid stylet lab was on the IB exam. So aphid stylets, it's a way of getting uncontaminated sap from the phloem. So an aphid, if your parents have rose bushes, when spring hits and those roses are just about to bloom, you're going to get these green bugs called aphids, and they like to eat the buds of the rose. These aphids actually like send like a little needle, proboscis, nose piece into the phloem, and they suck out the sugars. And we can have our aphids sucking on a plant, get our aphid out, and then break off that stylet, that proboscis, and measure the sugars that are in that little piece. Yes, it kills the aphid, which is why we don't do this as a lab in IB. So we remove the stylet, we analyze the contents or the sugars. Another way that's maybe a little more friendly to our bugs is through radioactively labeling carbon dioxide. So we can label the carbon dioxide, the plant absorbs the carbon dioxide, utilizes it for photosynthesis, and turns that carbon dioxide into glucose. And then we can trace through that radioactive carbon dioxide where that glucose is going. Pressure flow hypothesis here. It involves the active transport of sugars and the passive transport of water around the plant. Sugar is loaded at the source through active transport, which causes the water to enter through osmosis. Increased water in the sieve tube causes an increase in hydrostatic pressure, which causes movement of the sap. The sugar is removed at the sink and transformed into starch or used by the plant through active transport. Xylem then recycles the excess water. If you look at this, you should be able to figure out what the osmoter is the source. So this is your osmometer right here. Which one is the source and which one's the sink? So the source is here and the sink is over here. So the source is where it's brought in and the sink is where it's going. So what part of the plant is represented by A? Well, if we're talking about sugars, A would be the leaves and B would either be the root or the part of the plant that's growing. Here's the phloem location. Remember, the phloem's always on the outside and the xylem's always on the inside. This picture here or this picture will show up on a lot of tests, and it will have an arrow pointing and it'll say which one's the phloem and which one's the xylem. So make sure you understand this picture and this picture. And yay, we're done.